Okay, I think we can go ahead and get started for this evening. So, good evening and welcome to our webinar, Patient Powered Progress, Turning Tissue Donations into Discoveries through Pattern.org. As some of you may know, but just in case you don't, we're the Rare Cancer Research Foundation, a nonprofit that is dedicated to curing rare cancers through strategic investments and innovative collaborations that facilitate effective research and accelerate the deployment of promising therapies. That's a lot, but you'll get to understand what that means here soon. As we start, I just want to provide a few details for you. Throughout the call, your camera will be turned off to reduce distractions, but you should have the ability to access the chat to leave your questions. So please feel free to leave your questions in the chat on a rolling basis, and we will answer them at the end of the presentation. Also, at the end, I will provide a Google form link in the chat where you're welcome to leave feedback and any questions you may think of later, and we'll do our best to get back to you as soon as possible. So without further ado, I want to introduce Barbara Van Hare, who is our head of partnerships for the Rare Cancer Research Foundation. Barbara's been with RCRF since its inception, and she currently leads all of the organization's patient advocacy and institutional relationships, as well as manages Pattern.org's tissue collection process. So Barbara, feel free to take it away.
Thank you so much, Jenna. Appreciate it, and welcome everyone to our webinar. I think a lot of you may know me, but for those that don't, Jenna said that I've been with RCRF since the beginning, and delighted to still be here and talk to you about tissue donations through Pattern.org. So the Rare Cancer Research Foundation is a 501c3 nonprofit. It was founded in 2013 by a rare cancer patient by the name of Mark Labs. He was diagnosed with ocular melanoma at the young age of age 28. And as he researched his disease, he found out what it had in common with almost every other rare cancer is that there was limited research that had been done in the previous 25 years, and that the barriers causing the lack of research were similar across all rare cancer indications. So he created RCRF to eliminate the barriers that were slowing research. And you'll see on the screen our mission, which Jenna discussed earlier. So some of the key issues preventing research from moving faster for rare cancers is that there are limited patients at any single institution. That even includes the large academic centers. We're asked by some of the largest centers in the country if we can ship additional tissue to them because even they don't have enough. Some institutions store tissue at their institution. We're thrilled when they utilize that tissue for research, either at their institution or at others. But in many cases, that rare cancer tissue is stored and is never used, and or it is stored in a way that it's very limited in what it can be used for. Because there are few rare cancer patients for any particular indication across the country and across the world, many researchers lack access to that tumor tissue. And because they lack access to tumor tissue, many cases there are no or very few models of that particular disease. And if there are models, if a researcher has made some models, many times they're locked in that particular researcher's laboratory or that researcher's institution, and therefore aren't available to other researchers throughout the world. And patients can become partners in research to help solve all of these issues above. So our flagship programs, some of you may have heard of Pattern.org. It is an initiative of RCRF that has been around for about the past 7.5 years. It was the first program that we created where patients were really given the ability to drive research progress for their disease. All of our programs, besides the fact that they can empower patients to drive this progress, they allow researchers to find meaningful patterns in the cancer tissue and data, hence why the name Pattern or Pattern.org is used in all of our initiatives that involve where patients can help the research community find patterns. So Pattern.org will describe first, and then later in the presentation, I'll talk to you about our newest pattern initiatives, our data commons and biobanking. So, pattern.org is a revolutionary platform that empowers patients, regardless of where they're being treated in the US, whether it's a large academic center or a small community hospital. They can donate tissue samples, it would be excess tissue, and their clinical data directly to researchers, therefore helping bridge that gap between the patients and scientists and eliminating the barriers that are preventing the progress. So patients themselves have really almost all the power to help make a difference. Fresh tissue donation is historically what pattern.org has specialized in. And one of the key aspects of fresh tissue is the fact that it is fresh living tissue directly from surgery or a fluid drain. And it is still viable when it gets to the lab the day after surgery. And one of the things that fresh tissue is known to be used for is to help create disease models, such as cell lines or organoids, that enable scientists to better understand the biology of the disease and can hopefully help test and identify ways of diagnosing the disease and preventing the disease, and test and identify new therapies and potential cures. And if you'll see the image to the right of the screen, that is actually a pseudo-colored desmoid tumor cell line image from a patient that donated tissue through Pattern several years ago. And this image is hanging in the walls of the Koch Institute of MIT, where it won a 2023 Image Award. So how does pattern.org work? Well, first, patients learn about pattern.org and the fact that they can go onto the website and learn about research studies for their disease. Once they put in some basic information about themselves, they are matched to a study that needs their tissue, and the patient can then consent online. I will note here that pattern.org is in the process of being upgraded, and within the next month or so, patients that are interested in donating, if there's more than one study that has an interest in their tissue, they can let us know their preferences of how they would like that tissue shared. Once the patient consents online, then immediately the pattern.org team is notified, literally within about a minute of that consent. We then start working with the hospital where the patient is being treated. We'll coordinate with either their surgeon, the surgeon's office staff, the pathologist, the oncologist, whoever we need to at that particular institution for that particular patient, for that particular surgery or fluid drain, and get the hospital to agree to release the tissue. We then send it in a kit, which I'll show you in a minute, so that they can easily put the tissue into the kit and then overnight it at our expense to either the research project that it's going to or to our biobank. So here's a sample of one of our kits. Normally, what we ask for when a patient consents from the hospital is to collect excess tumor tissue. And what I mean by excess is tumor tissue that is not needed for clinical care of that patient. First and foremost, any tissue that's needed for clinical care, absolutely that should stay at the hospital. And if there's not enough for us, that's okay, because we want the patient to get whatever care they need. But if there is excess tissue, usually the surgeon or the pathologist will put tumor tissue. If some normal tissue is collected, they'll put some normal tissue also in one of the vials, and then also put blood in the blood tube. We use a nanocool, which you'll see there, kind of the silver top, which will keep the tissue viable at 4 degrees, which is basically refrigerator temperature. We use the nanocool because that is an easy way. All the pathologist or the surgeon has to do is push a button to activate it, and then it will start cooling that kit. Once the tissue is collected, those kits are overnighted from that procedure at that hospital to either the designated lab or to our biobank. We'll talk about that in a few minutes. And We normally ship through FedEx overnight delivery, so it arrives at that lab by 10:30 the following morning. We're often asked, so where does pattern.org fit in versus what the partner researcher or institution does? So Pattern really focuses on that patient consent and the tissue donation and the work that we do with the hospital to get them to release the tissue. Um, we collect that tissue and we also ask patients that are donating through Pattern to release their medical and clinical records to us. Both the tissue and the data from their medical records are de-identified, so the researcher or the institution or the biotech receiving that tissue does not know who that patient is. Then the partner research institution, usually what they do if it's living tissue is they try to create models. They may perform various genomic analysis. They may do single cell sequencing, and they may do other testing on that fresh tumor tissue. And then they publish and make accessible any and all of the results from the research that they did with the tissue. So if they were able to create a model, that model, in order for them to receive tissue through us, needs to go in the public domain. Any data that is generated from that tissue also needs to go into the public domain so that those results are shared broadly. As I mentioned earlier, the foundation was founded in order to break down the barriers. And so the more researchers that have access to these tools, the better it is for the patient community. In the case where we might be working with a biotech or pharma, if there's proprietary information that is generated from the tissue, we may make a carve out for that proprietary information, but the non-proprietary information does need to go in the public domain. Our foundation partners are critical to our success. This is just a list of some of the partners that we work with. Primarily, they have historically been the groups that let patients know about the opportunity to consent to research. And it's a win-win for everyone because the research is being done on behalf of the individual patients, but also the patient communities that are represented in these foundations as well as many others. We collect tissue from anywhere in the US, including Hawaii and Alaska. To date, we have collected from 125 different hospitals across 39 states, and that number is growing. We have research partners. The ones listed on this page are ones that are current partners or soon to be partners, probably within the next month or so. There's an additional, I'd say 13 or 14 that we're in various stages of negotiation with that we also hope to have onboarded within the next 6 or so months. To date, over 65 research models have been created from patient tissues that have gone through Pattern, as well as countless data sets that have been generated from the tissue that are hopefully helpful, critical building blocks that will lead to new therapies and potentially cures. So for live tissue donation, which is historically what we have specialized in, as I mentioned, we will only take excess tissue. Any tissue that goes through pattern.org will not affect clinical care. Again, if a doctor doesn't think that there's enough tissue, then we will not collect from that sample. The normal sample size that we're asking for is about a 0.5 centimeter cubed. We can accept less, we can accept more. It will vary greatly depending on the size of the tumor that the patient may have. We get asked a lot about biopsies, if we can take tissue from biopsies. Usually the amount of tissue coming from biopsies is very limited, and usually there's not excess tissue. It's not to say that we can never take excess tissue from biopsies, but really a resection or a fluid drain is the best to ensure that it's excess tissue. For certain cancers, depending on the research that's being done, and we will collect it if the researcher thinks that pleural effusion, which is fluid that develops around the lung, or ascites fluid, which is usually abdominal fluid, that usually occurs in late stage cancers. Many times that has a lot of cancer cells, viable cancer cells. And so depending on the research study, if they're interested in having us collect that, we will do that. We ask that patients consent prior to the procedure because that's really the only way for us to get living tissue unless other agreements are made with a medical institution up front. We ask for 7 days in advance. We know that's not always possible, but the reason we ask for 7 days is we actually have to get a hold of the clinical care team and usually the pathologist at that hospital. Many of them don't work on weekends. Once we get a hold of them, we have to get agreement. Sometimes that has to go up to the tech transfer office for them to agree to release the tissue. And so by the time we get through all of that work, then we will overnight a kit, and we would like that kit to be there at least 1 day, if not 2 days before the surgery, just to make sure there are no delays by FedEx. I will say, even though ideal is 7 days, we have in many cases collected from patients that consented just a day or two in advance in order to make it happen. Patient privacy is very important to us, so the tissue and data is all de-identified, and our data is encrypted on a HIPAA-compliant server with very restricted access. A patient can revoke consent at any time if they decide they no longer want to be in the study. The live tissue donation to research is at no cost to the patient and is for general research only, which means that unfortunately we're not able to give the patient any information about what happened with that tissue. We're looking at different ways that that may change in the future because we would like to give information back to that patient if we can. There are currently some novel projects that are being investigated where there would be a way to keep the patient information de-identified, but at the same time, let the patient know if a cell line, for instance, was created or not. So more to come on that in the future. We at RCRF feel very strongly that genomic sequencing is a great asset for rare cancer patients. And so we have a program where we try to help patients get access to molecular diagnostic and treatment information through various sequencing providers, such as Boston Gene, Tempus, Caris, really any of those that are out there, we can work with. We have the forms for most of those companies, and we will work to try to help the patient understand if they have never heard of sequencing, and in some cases, even their oncologist don't know the value of sequencing. So we will work with the patient and or the clinician to help explain why it might be helpful, help them complete the forms if necessary. Any of those samples, we would try to get a sample from that same surgery from tissue that's stored at the institution. Although we could take it from tissue that has been sent through us, ideally we will work to get it from the institution and have it sent to one of these CLIA certified labs, which basically means if it's CLIA certified, then it can be used to help inform clinical care. We would recommend whole exome sequencing and RNA sequencing. Reports do come out that go back to the oncologist that then they can discuss with the patient. Most all of these companies do have genetic counselors available if that's of interest. And in most cases, insurance will cover the cost. In the times that insurance won't cover it, many of these companies have funds that may assist with the cost. So in most cases, not all, usually if there is a cost, it's $100 or less. If the patient decides to go this route, they also have the ability to share the data, the raw data, as well as the report with RCRF. Any of that data that they share with us would be de-identified, and the patient would have access to that raw data if they would want it, either now or in the future. And the value of sharing this data back is kind of the big buzzword in cancer research today is AI. And if you don't have data sets, AI can't really do its job because it doesn't have enough data to model itself on. So we would encourage the sharing by any patients if they're willing to share with us. So what happens to a patient if they don't have an upcoming surgery or a drain? Is there anything that they can do to help participate, to help research? And the answer is absolutely yes. As of several months ago, RCRF is now accepting stored tissue samples. We have some research projects, depending on how the tissue has been stored, that can use those previously stored tissue samples. And patients, even if either they don't have any excess stored tissue sample or don't have any tissue at all, they are welcome to contribute their medical data with or without the tissue samples. Pattern.org, as I mentioned, is in the process of being upgraded. Once that upgrade is finished, patients will be able to consent for either sharing their stored and or their medical data only through pattern.org. In the meantime, it's a little bit of a manual process for us, but we're happy to talk to any patient and make it as easy as possible to have them do their donation. And if they aren't interested in sharing stored samples or their medical data, even if they just want to tell other patients about RCRF and what we're up to, that would be greatly appreciated. New for us within the past 6 or so months is our Pattern Biobank. We are partnering with a third-party company called Precision for Medicine. It is actually a contract research organization, also known as a CRO. It's an international company, and one of their specialties is biobanking. They're CAP accredited, GLP certified lab. They have very standardized workflows. They have extremely good security and, most importantly, backup generators to make sure that these samples are safe and secure. We work closely with them on each sample that may come in. So we work on the biologistics to get the sample in, how that sample is processed, and how it is stored. We maintain the inventory management and the sample distribution. And the type of tissues that we store currently are normal and tumor tissue, blood samples. We can both do regular blood collection tubes or Streck tubes. Streck tubes are known to be particularly effective for circulating tumor DNA. They can be stored, the tissue can be stored as FFPE, which is in formalin, as flash frozen tissue chunks, minced tissue in cryoprotectant media, and digested tissue in cryoprotectant media. And basically, the two options in the cryoprotectant media keeps that tissue living even though it's in a very deep freeze. From the blood, we can store PBMCs, plasma, double spun plasma for cell-free DNA. And if a research partner, whether that be a foundation or a researcher at a lab, has other ideas on how they would like tissue stored or need to be stored, we are definitely open to discussing those opportunities. We also have the Pattern Data Commons that isn't quite prime time yet. We are still kind of in the friends and family mode and hope to launch the project by fall. But our goals of our Data Commons is open, responsible sharing of rare cancer data to foster collaboration, drive innovation, and improve patient outcomes. What's different about our Data Commons, besides the fact that it focuses on rare cancer, is that it's built by data harmonization experts who were at the Parker Institute and helped build their Data Commons. It can accept any biological or clinical data, including imaging. Data in any format can be accepted, and they'll harmonize it with the other data. And we can accept data sets from patients that consent through Pattern or data sets that consent through other places and want to share their data with us. We can accept data sets from institutional data registries or foundation registries. We're willing to work with whatever rare cancer group is interested in sharing their data with us. It is open access, so there's no outdated technology or restrictive agreements. We are not about IP. Um, Researcher access would be across the field, meaning researchers from various academic and other institutions, biotech, pharma, and we will implement the highest standards of privacy and security. Again, this also helps enable AI and the future of precision medicine. And as our team that's creating the data commons would say, every data set becomes a step towards a breakthrough. So overall, what pattern.org and RCF is empowering patients, identifying the patterns, and advancing research. So on the left-hand side, you'll see how patients can contribute to research and empower themselves through live tissue donation, genomic sequencing, stored tissue donation, data-only donation, and custodial tissue donations in the biobank. And I'm going to stop here to talk a little bit about custodial tissue donations because that is a relatively new program for us. Patients that are interested in storing their own tissue, we now have a program where they can do that. It is a different consent and protocol than what we use for the research donations, where the patient can pay to store their tissue, and it can be used either for research projects they're interested in or for clinical projects that may be able to help their care. For more information on that, reach out to us. The prices vary based on what exactly the patient wants to share with us and what they would like to do with the tissue. So we would set up a meeting with the head of our biobank, discuss the details, and work individually with each of those patients. Also, I should say that for any foundation that's interested in starting a collection of tissues for their particular disease, we also can do that. It's not exactly custodial tissue. It would be more research, but the foundations would own those tissues and decide how they should be best utilized. All learnings and data are available to rare cancer research in our pattern data commons, and then researchers would use those tools to help research be accelerated through models, data sets, precision for medicine, which would help individual patients, and pathways to cures. And as I alluded to, we partner across all stakeholders. So whether it's a patient that's interested in donating tissue to us, a foundation that wants to work with us, either to get tissue collected to go to a particular research project that they want to help support, or they want to store different tissue types in our biobank. If clinicians want to work with us in helping to send their patients to pattern.org, if researchers want to work with us either to receive tissue through us or work on various projects that we may be involved with, we definitely welcome that. If academic institutions want to partner with us, we're currently working with a fairly large academic institution that wants to start having all of their patients be able to consent through pattern.org, and then they would share that tissue with us, and we'd have kits at that institution, so as surgeries came up, they could be collected there right on site. And we're more than willing to do that with other academic or small community and regional hospitals. We've recently had interest from several regional groups that want to tell all of their cancer patients about us and start sending more tissues to us that way so that they can help research. Also, we're interested in work with biotech and pharma. So if anyone would like more information or interested in partnering with us, feel free to reach out to me at barbara@rarecancer.org. And now, time for Q&A. And I will turn it over to Jenna to ask any of the questions that may be coming through the chat box.
Sure, absolutely. So we have several great questions. Our first one is pattern.org only in the US? I'm located in Canada.
So currently, we are only collecting in the US. Canada requires a separate consent and protocol. We are interested in going to Canada. I would probably say that would be later this year or next year. We, a number of years ago, did get some fluid samples out of Canada. We're a little concerned about the fresh tissue coming across the border and the delays that might be held there, but we're definitely willing to try it. So if you want to send us your information, we'll let you know as soon as we're live and able to collect from our friends across the border.
Great. Our next question is tissue donation safe and how is it regulated?
How is tissue donation safe? Yes, in our case, tissue donation is safe because we are not taking any tissue that wouldn't automatically be taken from the patient in surgery. So as the tumor, that's why we're taking excess tissue. So we're not taking tissue just to take tissue, we're taking basically leftover tissue. As far as regulation, there are a lot of regulations about informed consent to make sure the patient knows what they're consenting to. And that's one of the values of having our consent online because a patient can take as long as they need to go through the consent online. And there's even a chat box if they have questions while they're going through the consent, or they can read through page by page, stop, go do something else and come back to it the next day to continue go through it, and or print it out and read through it. Plenty of time to make sure they understand what the tissue may be used for. There are also regulations in terms of shipping tissue across the country. We follow all of those regulations. And then there are different regulations if there's any post-mortem tissue collected. Um, we historically have not gotten very involved with post-mortem tissue because of all of the potential complications, but there are also regulations for that. And then, of course, there are international regulations, but currently we aren't collecting any tissues from overseas. Um, we are looking at potentially maybe not collecting data from overseas, but sharing the data that we do collect overseas and making sure that we would be compliant with any regulations, either here in the US or in the other countries.
Okay. What type of patterns do you look for? Specifically, do you look for genetic mutations?
So the genetic mutations is really what the genomic sequencing is about. And that's why we're encouraging patients to get that because with rare cancers, quite a number of them, the vast majority, not a lot is known about these different cancers. And so one of the reasons we're recommending whole exome sequencing is there may not be a lot known about different mutations for that cancer today, but that could change in the upcoming months or the upcoming years. And so that's what we encourage patients to do if they're willing. As far as the other patterns, that is a better question for an actual researcher when they're doing single cell sequencing and all the other types of tests that they do in terms of what exactly they're looking for in the tissue. And that will probably vary based on the research project and what they're trying to learn from it.
Okay. Would blood samples be able to be used for circulating tumor DNA?
Absolutely. As long as the blood is put in the correct blood collection tube, STREC, STRECK is one such tube. We keep those on hand. Um, and that is kind of I think the gold standard they would say today for circulating tumor DNA. There's something in the tube, I am not a scientist, but that keeps the blood best for ctDNA. And something that I neglected to mention earlier, if there are projects that just need blood collected and not actually tumor tissue, we have the capability of to do that. We have actually done that for quite a few patients, and we have actually a blood collection kit that just has either Streck tubes or what are called purple top EDTA tubes, which are more standard blood tubes. And we can configure those blood collection tubes in almost any format. We've been asked actually by a number of rare cancer groups as well as rare disease groups if we could start doing blood collections for their patient community, and the answer to that is yes, we can. Our protocol and consent allow for that.
Great. The next question is, although I know there's a need for all rare cancer tissues, are there types of rare cancer tissues that are currently being sought after by your affiliated researchers more so than others?
There's quite a list. There is a lot of interest in ocular melanoma, virtually all types of sarcomas, um, head and neck cancers, rare kidney cancers. There's going to be a project for some rare lung cancers. Um, we're getting more and more requests almost weekly for different types of rare cancers. And that's one of the reasons that we started the biobank, was more what we're calling for short-term storage, not long-term storage. The key was to make sure that we had a place for rare cancer tissues to go, that even if we don't have a research project today, we either might know of one coming in the future, or we can be on the lookout and actually seek out researchers that might need that tissue so that it can be effectively utilized. So our biobank isn't like a lot of biobanks that we would like to have the tissue stored for years. We'd like it, basically, we say tissue in and tissue out. We want it to get to the researchers so they can do all the great things that they do so new therapeutics can be developed.
Great. Our next question is, have you partnered with the Count Me In project?
So we are good friends with the Count Me In project. The difference between the two projects is Count Me In has focused on select different indications, and their primary goal is to do genomic sequencing on those particular cancers and share that data broadly. And as I mentioned, we feel that that is a very critical thing to do, and we love the fact that they've been doing it. All of the tissues that they collect are stored tissue, primarily those that have been in formalin. I don't know if they've done any flash frozen tissue. They have not collected fresh tissue, and that's one of the key differentiators. They're doing sequencing from stored samples, and we historically have really focused on fresh tissue, and we will continue that emphasis, but now are also opening the door for those patients that would like to share their stored samples.
Okay. Will there be an opportunity for international collaboration for the various systems under the umbrella that is RCRF?
We hope so. We definitely hope in future time that we can collaborate with people from all over the world. There are various regulations on data and tissue and sharing. And in some cases, like on the tissue, it can vary by country. And in the data, I think the European Union has certain regulations and other places have others. So that definitely would be in our long-term plan, just not in the immediate future.
Okay. Is there ever a problem with the institution not honoring the patient's consent to relinquish tumor tissue?
Unfortunately, yes. Um, we have encountered a number of institutions because historically, the way the institutions, particularly academic institutions, have run their institutional safeguard and standards is once the tissue comes out of the body, then the institution feels it's theirs. And in many cases, patients that may be signing that consent there at the hospital. In my case, when I've had surgery, I literally signed the consent when they were getting ready to, wheel me into the operating room. And the tissue they feel is theirs and they don't want to release it, because either they want to keep it for researchers that are there at their own institution, which is awesome if they are going to use that tissue and actually do research with it. Um, and they're also, in many cases, concerned about intellectual property. So in a number of cases, they have told us, well, we'll only release the tissue if we negotiate what's called a material transfer agreement, where they want to negotiate intellectual property rights, and it can take months, if not years, to get those different agreements signed. We're not interested in the intellectual property rights. We're interested in getting the tissues to the researchers that are going to do great things to help patients and actually help the entire rare cancer community. Because if the researchers can find things with these tissues, not only does it help the patients, which is most important, but it will help the clinicians better treat the patients. It will help pharma know what drugs to create. It will help the whole ecosystem function more effectively, and find new therapies and cures, for those that need it most. But I would say overall, in most cases, we are able to get the institutions to release the tissue and honor that patient's wishes.
Okay. Is there a future collaboration with the NIH at some point?
Ah, that's a really good question. We have been in discussions with various groups at the NIH. We've been a partner in a couple of their projects there. Um, there's a lot going on at the NIH today, so I don't think in the next few months probably is the best time to try to start a new partnership, but definitely something that we would like to do once things calm down a bit, at NCI and NIH.
Okay. Can frozen or paraffin fixed samples be analyzed?
Yes, depending on the testing that's going to be run, they can. They're used for different things. As I mentioned, like fresh tissue is really good for like model generation, and some feel that it's superior for single cell sequencing. But like genomic sequencing, is great out of formalin fixed tissue. Flash frozen can be used for certain things.
Okay. Do we have many neuroendocrine tumor samples?
Um, I don't know if that question is for in our biobank. Do we have many of those? A project that we have supported for since we created pattern.org is the Cancer Cell Line Factory at the Broad Institute. And we have sent them quite a few neuroendocrine samples. I think we have a few in our biobank is relatively new, so we don't have many samples yet. But if you know of any neuroendocrine patients that are interested in donating either stored samples or fresh tissue samples from upcoming procedures, we would love to hear from you or those patients.
Great. Will the tumor tissue material be able to be developed into cell lines for researchers to access? And how are the tumors authenticated?
That's a really good question. So, yes, I'm going to use the example of the Cancer Cell Line Factory at the Broad Institute, where their whole focus is on creating these living cell lines or organoids, which are basically living models of that particular cancer. Um, if they're able to create a cell line, then they have deposited them into a public biorepository called ATCC, which is in Manassas, Virginia. And those cell lines are sent there, and then they are what is called expanded, meaning they're increasing the number of those cell lines so that they can sell them to research partners across the world. Any of the cell lines that go there or really to any of those public biorepositories go through stringent validation studies, to confirm that they are the cancer that they're purported to be and representative of that cancer in humans.
Okay, I have asked if anyone has any other last minute questions. So we'll see if anyone drops anything in here shortly. But otherwise, I'm going to drop the link that I mentioned earlier in the chat for the feedback form for everyone. I'll do that right now. Okay. And if no one has any other questions, I think that's good. Barbara, what do you think?
If anyone wants to discuss anything further or has any questions, feel free to contact me directly, Barbara at rarecancer.org. Would love to talk to you or any of the associates you know that may have questions or may be interested in partnering.
Wonderful. Thank you so much, everyone. Have a great evening.
Thank you.